Introduction. Welcome to today's lecture on the fascinating world of protozoan parasites. Protozoa are unicellular eukaryotic organisms that play a significant role in both human health and ecological systems. Throughout this lecture, we will explore various topics concerning these parasites, ranging from their molecular mechanisms of drug resistance to their impact on autoimmune diseases and climate change. By gaining a comprehensive understanding of these subjects, we can shed light on potential therapeutic targets, diagnostic methods, and strategies for infection management. Now, let's dive into the world of protozoan parasites and begin our exploration with the first topic. Protozoan parasites have evolved multiple mechanisms to withstand antiparasitic drugs, making them less effective. These mechanisms include overexpression of drug efflux pumps, mutations in drug targets, and upregulation of detoxification enzymes. Understanding these molecular mechanisms can lead to the identification of therapeutic targets and the development of new drugs to combat drug-resistant protozoan infections. Certain protozoan parasites, like Toxoplasma gondii and Leishmania spp, might contribute to autoimmune diseases. Infection can lead to persistent immune activation, dysregulation, and production of pro-inflammatory cytokines, causing chronic inflammation and tissue damage. Understanding this link is critical for targeted interventions. Climate change has a global impact on protozoan parasites, affecting their distribution and transmission. Rising temperatures, changing rainfall patterns, and ecological shifts can expand the range of vector species and create suitable breeding sites, increasing the potential for disease transmission. Understanding these interactions is important for predicting and managing protozoan parasite-related diseases in a changing climate. Examining the genomic diversity and evolutionary history of protozoan parasites provides insights into their adaptability and survival strategies. High levels of genetic diversity and rapid adaptation are common traits among these parasites, allowing them to evade host immune responses and develop resistance to antiparasitic drugs. Techniques such as whole genome sequencing and comparative genomics have unveiled the immense genetic variation within and between protozoan parasite species. Understanding the genetic mechanisms responsible for these adaptations can aid in the development of interventions that target essential pathways critical for parasite survival. Protozoan parasites can contaminate food and water sources, causing foodborne illnesses in humans. Parasites such as Giardia lamblia and Cryptosporidium parvum are responsible for numerous outbreaks worldwide. Contaminated food items, including fresh produce, seafood, and improperly cooked meat, can serve as vehicles for transmission. Additionally, waterborne transmission, especially in inadequately treated water sources, poses a significant risk. Understanding the prevalence, transmission routes, and methods for detecting these parasites in food and water is crucial for implementing preventive measures and ensuring food safety. Co-infection with multiple protozoan parasites can significantly impact disease outcomes. The interaction between different parasites can result in synergistic effects, altering disease severity, treatment response, and immune responses. For example, co-infection with Plasmodium spp. and HIV has been found to exacerbate the clinical manifestations of both diseases. The underlying mechanisms driving these interactions include immunomodulation, competition for resources, and the activation of shared pathways within the host. Recognizing the complexities associated with protozoan co-infections is necessary for developing appropriate diagnostic methods and treatment strategies that address these combined infections effectively. Efficient and accurate diagnosis of protozoan parasitic infections is important. Traditional methods like microscopy and serological tests have limitations. New molecular techniques like PCR and LAMP improve detection and identification. Point-of-care tools help resource-limited settings. These advancements aid in timely diagnosis, treatment, and surveillance. In a fascinating twist, certain protozoan parasites are being explored as potential biocontrol agents to mitigate the impact of disease vectors and agricultural pests. For example, the protozoan parasite Amblyopspora spp. has shown promising effects against mosquito vectors that transmit diseases such as dengue and malaria. Similarly, the use of microsporidian parasites against agricultural pests like Helicanverpa armigera has gained attention. These protozoan parasites can infect the target vectors or pests, reducing their populations and limiting disease transmission or crop damage. However, careful evaluation of their safety, environmental impact, and effectiveness is necessary before implementing such biocontrol strategies. Protozoan parasites like Blastocystis spp, Entamoeba histolytica, and Dientamoeba fragilis can alter the gut microbiota, causing dysbiosis and potentially leading to diseases like inflammatory bowel disease and metabolic disorders. Understanding these interactions can help develop interventions for a healthy gut microbiota. Host genetic factors strongly influence an individual's susceptibility or resistance to protozoan parasitic infections. Variants in the immune system genes affect the infection response, impacting disease outcomes and treatment effectiveness. Research on these factors helps develop personalized medicine approaches, such as identifying high-risk individuals and tailoring treatments based on genetic profiles. The immune system is crucial for controlling and eliminating protozoan parasites. Immune cells like T cells, macrophages, and natural killer cells recognize and destroy infected cells, clearing the parasites. Understanding this interaction can help develop immunotherapeutic strategies, such as cytokine administration or immune checkpoint regulation, to enhance parasite clearance. Therapeutic vaccines can also provide long-term protection against protozoan infections. These strategies show promise for improving infection management and reducing the impact of protozoan parasitic diseases. As we conclude our exploration of protozoan parasites, we have gained insights into their biology, impact on human health, and potential therapeutic interventions. Thank you for attending today's lecture.